Well, hello everyone. Today I'm excited to share with you one of my projects, which investigates the role of ceramide in the progression and severity of allergic asthma. So asthma is one of the most common airway diseases, with an estimated 339 million people having it across the world. The most common form of asthma is allergic asthma, with clinical features of hypermucus secretion, inflammation, and airway hyperreactivity, all of which reduce the amount of volume available for airflow, and this is what causes the difficulty in breathing. Now, typically, these symptoms can traditionally be managed with corticosteroids or bronchodilators, but for cases where there's more severe, this can lead to higher fatalities. And so, a common interest between clinicians and researchers alike is to understand more the mechanism underlying allergic asthma. But to give a brief introduction with what we already know, we know that during allergic asthma, the lung epithelial cells are exposed to some type of allergen, in this case, house dust mite. And this initiates the recruitment and maturation of dendritic cells, which then are going to move into the secondary lymphoid system. And it's there that they're going to present a piece of the allergen to naive T cells and induce a Th2 or T helper 2 polarization. And T helper 2 cells is what's going to orchestrate the classical Th2 inflammation, such as the production of IL-4, 5, and 13. And then we see inflammation predominantly through eosinophils, airway hyperreactivity, and airway remodeling. And so for my project, we utilized a mouse model that recapitulates a lot of the clinical phenotypes that I just went over. And so to do this, I used a C57 Black 6 mice that's given a total of 10 intranasal injections of house dust mite over the span of about 2 weeks. And on day 15, the mice are sacrificed, and I was able to measure levels of different immune cell populations in the bronchoalveolar lavage fluid, or BALF. And so what we saw was that the HDM challenged mice all had a significant increase of a variety of different immune cells, especially eosinophils, which, as I mentioned before, are the predominant inflammatory immune cells in allergic asthma models. We also used the flex event respiratory system to take in vivo measurements of lung resistance, which is a measure of airway hyperreactivity, and saw that the house dust mite group had increased lung resistance. Interestingly, ceramide and the lung lysate of the house dust mite challenged mice also was significantly increased, specifically in the C16 and C24 species of ceramide, which are the prominent species in the lung. We also saw a striking induction of apoptosis, which we measured by levels protein expression of cleaved caspase 3. And in the lung lysate of house dust mite challenge mice, there was also a significant increase in oxidative species, which we measured as ROS using a general oxidative stress indicator. Lung sections from saline and house dust mice also confirmed our finding of increased ceramide and cleaved caspase 3 staining in the HDM group. But I also wanted to bring it to attention this increased co-localization that we also saw between the cleaved caspase 3 and the ceramide, which is in line with previous knowledge that we all know of ceramide being a potent inducer of apoptosis. But to actually determine if the heightened cell death and generation of oxidative species were in fact reliant on this increased levels of ceramide that we're seeing in my house dust mite model, we introduced 2 ceramide synthesis inhibitors during the final 3 days of HDM challenge. And these were myriosin and phenomycin B1, which you all are probably familiar with. And so what we saw was that the introduction of either of these inhibitors significantly reduced the levels of ceramide, this time measured in the BALF of our house dust mite groups, and also the dihydroceramides. I do want to bring attention, though, to myriosin, which was a bit more effective at reducing the levels of ceramide in comparison to the phenomycin B1, which may indicate that in this house dust mite challenge model, the source of ceramide could be coming more prominently from the de novo pathway versus the recycling. So what we saw was that the suppression of ceramide in production, impressively reduced levels of apoptosis. And we show this by tunnel staining of the different lung sections from the house dust mite group and the house dust mite groups that were given the different inhibitors. And you can see that there was a significant reduction in the tunnel staining for both groups. And then this result was also confirmed with, once again, Western blots for cleaved caspase-3. And we also saw in the lung lysate that levels of ROS were reduced when you introduced the muriocin or futamycin B1. And so from these results, we were able to conclude that the induction of oxidative stress and apoptosis in the HDM challenge mice were ceramide dependent. Another question that arose during the study was if the increased apoptosis observed in my HDM challenge mice was dependent on ROS. So as of you know, most of you probably know, ROS can be harmful to cells when it gets beyond the homeostatic levels, causing extensive cell damage and leading to cell death. So this offers two possible scenarios in my HDM challenge mice. So it could be that during HDM challenge, where you have that subsequent induction of Th2 response, an elevation of ceramide, the increased ceramide is inducing excessive production of ROS, and it's this ROS that's causing cell death or apoptosis. Or it could be that ROS production and the induction of apoptosis are both independently induced by elevated levels of ceramide. So to understand or figure out which scenario is going on, we collaborated with Dr. Joanne Cook-Mills of Indiana University, who utilizes a similar house dust mite challenge model, but in this case, her mice are also put on an alpha tocopherol supplemented diet. And alpha tocopherol is an isoform of vitamin E, which is an antioxidant. So this offered a great opportunity where we could reduce levels of ROS using the alpha tocopherol vitamin E isoform and see if the levels of apoptosis were being affected. And so first, once again, we needed to measure levels of ROS in the lung lysates of these different groups of mice. And in the first bar graph here, we can see that the HDM mice that were put on the isoform, the alpha tocopherol isoform diet, had significantly less ROS in comparison to just the house dust mite group, which is great. So we moved forward and once again measured the ceramide levels and apoptosis in the lung lysates. And we saw that although there was a trend for higher ceramide, statistically, there was no significant difference between house dust mite and house dust mite alpha tocopherol mice. And this was the same for the cleaved caspase-3 groups. And so we concluded that the elevation of ceramide by house dust mite and subsequent induction of apoptosis was independent of its effects on ROS. And then moving into some pretty cool data with human patients, we measured ceramide levels in bowel fluid collected from either healthy, non-severe, or severe asthmatic patients. And we detected significant increase in longer chain ceramide species, C20, C26, and C26-1, and the severe asthma groups. Now, this is particularly interesting because severe asthmatic patients are a subset of asthma patients that don't respond to traditional corticosteroid treatments. And so there's a lot of interest in the field in understanding the mechanism behind this more challenging disease. But what we do know is that most severe asthmatic patients tend to have more neutrophilic inflammation in the lungs. And so interestingly, in our data, we were able to see this positive correlation between the percentage of neutrophils in the bowel along with levels of C16 and C26-1 ceramide. And then we also measured a negative correlation between lung function, measured here by forced exploratory volume, and the same species of ceramide. And so this data suggests association between levels of ceramide and markers of severe asthma, that is poor lung function and neutrophilia. Similar results were demonstrated back in our mice models. And this time we were staining our lung sections from our house dust mite and our house dust mite ceramide synthesis inhibitor groups by using markers for neutrophils, either Ly6G in red and marked here with the white arrow, or MPO marked in green and white arrow. And so what we saw was that both ceramide suppressors were able to reduce the staining of both neutrophil markers. And then in a separate experiment, I took C57 black 6 mice and gave them one intranasal injection of just C16 ceramide. And C16 ceramide alone was able to increase significantly the number of neutrophils within the bowel. And so both of these results sort of support our hypothesis that not only house dust mite, but ceramide specifically can trigger the recruitment of neutrophils in asthma models. And then finally, to take a closer look at the effects of ceramide on neutrophils, we simplified our model to just two players, primary epithelial cells and neutrophils. And so lung epithelial cells were stimulated with either ceramide or house dust mite, with or without the ceramide production inhibitors, FB1 or muriosin. And the stimulation went on for about 20 hours. Next, fluorescently labeled neutrophils were added to the stimulated epithelial cells. And then we allowed them to attach for 4 hours. And then cells that were not attached were washed off. And then the cells that were attached were measured through a plate reader. And so our results showed that both ceramide and house dust mite stimulated epithelial cells had increased neutrophil attachment. And this effect was reversed when you introduce the ceramide inhibitors, sputamine B1 and muriosin. And so this data further supports the relationship between ceramide and neutrophilia, but also suggests that lung epithelial cells can be an important player as well in this story. So to conclude, my project showed that in both asthmatic patients and in our mice allergen asthma models, ceramide levels are increased. An allergen challenge also increases levels of ROS and apoptosis, which are both dependent on ceramide. In severe asthmatic patients, we saw that there was a positive correlation between ceramide and neutrophils. And we also demonstrated in vivo and in vitro models that ceramide ceramide induces neutrophilia and also neutrophil attachment into lung epithelial cells. And so ceramide may be the trigger for important biomarkers of severe asthma, such as Crayola bodies, which are just aggregates of epithelial cells formed from excessive apoptosis, or neutrophil dominant inflammation. And so with that, I would like to thank my PI, Dr. Sarah Spiegel, along with the members of the Spiegel Lab. I would like to thank everyone who collaborated with us in this project and all of you for taking the time to listen.